Hey everyone, welcome back to the channel. Today we're diving into something absolutely fascinating from the intersection of deep learning and computational biology, the Stanford RNA 3D folding competition on Kaggle. If you've been following breakthrough AI developments in biology, you might have heard about AlphaFold revolutionizing protein structure prediction. Well, today we're looking at something similar, but for RNA molecules, which are equally critical for life. I've got a detailed walkthrough of a notebook authored by Kaggle GrandMaster Moth. The notebook breaks down RibonanzaNet, a state-of-the-art architecture for RNA 3D structure prediction. Let's study this notebook together. Examine each component piece by piece, understand how they work together, and see why this matters for fields ranging from drug discovery to understanding genetic diseases. Whether you're a machine learning enthusiast looking to understand transformer architectures in a new context, or a biology researcher curious about computational approaches to molecular structures, this video has something for you. So let's jump right in. The Stanford RNA 3D folding competition challenges participants to predict how RNA molecules fold in three-dimensional space based solely on their sequence. This is an incredibly important problem because RNA's function is determined by its structure. When RNA folds incorrectly, it can lead to diseases. The notebook we're exploring today breaks down RibonanzaNet, a neural network architecture proposed by Shujun He and colleagues. As described in their paper, RibonanzaNet, Deep Learning of RNA Structure Through Dual Crowdsourcing. This architecture combines elements from RNA Digformer and Top Kaggle models to create a unified approach to RNA structure prediction. The notebook starts with the typical setup sections. It's worth noting that RibonanzaNet relies on standard deep learning libraries like PyTorch, along with specialized utilities like INOps for tensor manipulation. These sections ensure you have everything needed to run the model. Next, we have the configuration section, which defines the model's hyperparameters. The notebook uses a YAML configuration file with various parameters like learning rate, batch size, number of layers, and so on. The default setup has a learning rate of 0.001, a batch size of 4, and 9 encoder layers. The configuration also sets up options like whether to use triangular attention and establishes the pairwise dimension size of 64. Think of this section as the control panel for the entire model. Tweak these settings, and you can balance performance against computational efficiency. The utils section provides helper functions that will be used throughout the implementation. There's a config class to manage hyperparameters, some utility functions with names like default and exists, and functions for initializing weights and loading configurations from YAML files. These might seem like minor details, but they're essential for keeping the code clean and maintainable when you're working with complex neural networks like this one. Now we get to the neural network components, starting with specialized dropout implementations. You're probably familiar with regular dropout as a way to prevent overfitting, but RibonanzaNet uses some specialized versions. There's a custom dropout class that can share the dropout mask along specific dimensions, plus variants called dropout rowwise and dropout columnwise that apply dropout across rows or columns. These specialized implementations help regularize the model in ways that respect the 2D structure of the pairwise representations, important when you're modeling interactions between nucleotides. Next, we encounter the misactivation function, which is a smooth, non-monotonic activation proposed by Diganta Misra in 2019. If you're used to ReLU, which is the standard activation function in many networks, MIS offers some advantages. It doesn't suffer from the dead neuron problem that can happen with ReLU, and it often provides better performance on deep learning tasks. The implementation is surprisingly simple. It's just x multiplied by the hyperbolic tangent of the softplus of x. This creates a smooth output that helps gradients flow better during training. Generalized mean pooling, or GM for short, is a learnable pooling operation that's more flexible than traditional methods. It's a clever way to generalize both average and max pooling into a single operation. By adjusting a parameter called p, the model can adaptively learn the optimal pooling behavior. When p equals 1, it's equivalent to average pooling, and as p gets larger and approaches infinity, it becomes more like max pooling. This flexibility is really valuable in RNA folding models because different parts of the sequence might benefit from different types of pooling operations. Now we're getting to the heart of the transformer architecture. Scaled dot product attention was introduced in that landmark paper. Attention is all you need, and it's fundamental to how transformers work. The implementation takes query, key, and value tensors, computes attention scores by taking the dot product of queries and keys, scales them, applies softmax, and then uses these attention weights to create a weighted sum of the values. In plain English, this mechanism allows the model to focus on relevant parts of the RNA sequence when making predictions about each position, regardless of how far apart they are in the sequence. Building on what we just discussed, multi-head attention extends the attention mechanism by applying multiple attention heads in parallel. Instead of having just one attention mechanism, you have several working side by side, each potentially learning different patterns. It's like having multiple experts looking at the same problem from different angles. The implementation projects the input into multiple heads, applies attention separately in each head, and then concatenates and projects the results back to the original dimension. This is crucial for RNA folding because different attention heads can specialize in capturing different types of interactions between nucleotides. Here's an interesting challenge with attention mechanisms. They don't inherently understand sequence order. If you scrambled the positions of the nucleotides, the basic attention mechanism would give you the same result. That's why we need positional encoding, which adds information about position to each element in the sequence. The implementation uses the standard sinusoidal encoding from the original transformer paper, which creates unique positional vectors using sine and cosine functions of different frequencies. This helps the model distinguish between different positions in the RNA sequence and understand the relative ordering of nucleotides. The outer product mean module is inspired by AlphaFold 2 and computes pairwise interactions between elements in a sequence. It projects the sequence representation into a lower dimension, computes outer products between all pairs of positions, and then projects back to the final pairwise dimension. This operation is crucial because it allows the model to capture interactions between any two nucleotides in the sequence, which is essential for understanding how distant parts of the RNA might fold together in 3D space. The triangle multiplicative module, also borrowed from AlphaFold 2, is designed to ensure that predicted distances between residues make geometric sense. In the real world, if point A is close to point B and point B is close to point C, then point A can't be too far from point C. That's the triangle inequality principle. This module updates each pairwise representation by considering how those two positions interact with all other positions in the sequence, helping enforce geometric consistency. The module comes in two flavors, ingoing and outgoing, which aggregate information in different ways to update the pairwise representation. Triangle attention builds on the previous module, implementing an attention mechanism specifically designed for pairwise representations. It can be applied either row-wise or column-wise to capture different types of interactions in the pairwise matrix. This specialized attention mechanism helps the model focus on relevant parts of the pairwise representation when updating each position, further improving the geometric consistency of the predictions. Now we reach the conv transformer encoder, which is the main building block of the RibonanzaNet architecture. This layer combines several operations. Convolutional operations capture local patterns, multi-head attention handles long-range interactions, triangle multiplicative updates ensure geometric consistency, and outer product mean operations update pairwise representations. What makes this architecture special is that information flows bidirectionally between the sequence representation and the pairwise representation. The model doesn't just use pairwise information to update the sequence understanding. It also uses the sequence understanding to refine the pairwise information. This constant back and forth allows the model to continuously refine its understanding of both local sequence patterns and global structural constraints. In addition to the standard positional encoding we discussed earlier, RibonanzaNet also uses relative positional encoding. Instead of encoding absolute positions, this module encodes the relative distance between any two positions in the sequence. The implementation uses a one-hot encoding of relative positions from -8 to 8, which is then projected into the embedding space. This helps the model better understand the spatial relationships between different parts of the RNA sequence. Finally, we reach the complete RibonanzaNet model, which integrates all the previous components. The architecture starts by embedding the input RNA sequence, then initializes pairwise features using the outer product mean. It adds relative positional encoding to provide spatial context, then processes everything through multiple conv-transformer encoder layers. Throughout this process, information flows back and forth between the sequence and pairwise representations, gradually refining the model's understanding of the RNA structure. Finally, it outputs predictions for each nucleotide, representing their positions in 3D space. In the final technical section, the notebook instantiates the model with the configured parameters and runs a simple forward pass to verify everything works correctly. With an input batch of 4 sequences, each 128 nucleotides long, the model outputs predictions with a shape of 4, 128, and 2. This demonstrates that the implementation is functional and ready for training on RNA sequence data. The notebook concludes by highlighting that RibonanzaNet combines elements from various successful approaches to RNA structure prediction. By unifying features from RNA Digformer and Top Kaggle models, it creates a more powerful, self-contained architecture. The author also points to resources for fine-tuning and inference, encouraging readers to build upon this implementation for the competition or their own research. There you have it, a comprehensive walkthrough of RibonanzaNet, breaking down each component that makes this architecture tick. We've seen how it combines convolutional operations, attention mechanisms, and geometric constraints to predict RNA 3D structures from sequences. What I find most fascinating about this approach is how it mirrors our understanding of the physical process of RNA folding. Just as RNA molecules fold based on both local interactions and global energy minimization, RibonanzaNet captures both sequence patterns and structural constraints through its bidirectional flow of information. The implications of this work extend far beyond the Kaggle competition. Accurate RNA structure prediction could accelerate drug discovery, improve our understanding of genetic diseases, and even help develop new RNA-based therapies like mRNA vaccines. If you're interested in exploring this further, the notebook provides detailed code implementations and references to papers for deeper study. And remember, the best way to truly understand these concepts is to experiment with the code yourself. I hope you found this walkthrough valuable. If you did, please hit that like button and subscribe for more content at the intersection of AI and science. Drop your questions or insights in the comments below. I'd love to hear which aspects of RNA folding or deep learning architectures you find most intriguing. Until next time, keep exploring, keep learning, and keep pushing the boundaries of what's possible with AI. Thanks for watching.